Okay, great. Thank you very much, and thank you for ARM for inviting us back to share the Fibrocell story. And we are a public company traded on NASDAQ, so we will be making some forward statements today. So if you're not familiar with Fibrocell, we have a platform of rare skin disease therapies that are based on the autologous fibroblast. And today I'll be focusing mainly on our recessive dystrophic EB. You just heard about the disease, and I'll be also focusing on our main program, which is an RDEB. I will also touch a little bit on our other program in localized scleroderma. And just to move right to the platform, our main program, as I mentioned, is targeting type 7 collagen. I'll talk a little bit about the background of the disease and a little bit about the biology of the protein. Our main program is a phase 1/2 trial. We are now currently enrolling in the phase 2 portion and have already treated our first subject in phase 2. We have orphan drug, rare pediatric disease, and fast track designation for this particular indication. I'll be touching mainly on this today. But we also do have a second indication in moderate to severe localized scleroderma. This is a condition that, based on an autoimmune reaction, you have this thickening of the skin. It's an accumulation of collagen 1 and 3, and it's very debilitating. It deals a lot with not only range of motion issues, but in children, where it is prevalent, can lead to problems with development because of these sort of solidified limbs. So a lot of morbidity in this disease, and we just recently received fast track designation on this one from FDA just a couple weeks ago, and announced that we have now opened enrollment on this trial in a single center in Philadelphia area, and really excited to announce some new milestones for this one shortly. But as I mentioned today, I will focus on our platform and on the EB indication. And as you can see, we are an ex vivo modified cell program, and which brings together a fibroblast technology we've been developing since, through the 2000s to now, and along with lentiviral vector technology. And we believe that the cell here is acting as a delivery vehicle to carry these gene constructs to the area of concern. So these are localized treatments in the area of wounds, or in the case of scleroderma and these lesions, and brings the construct to produce a protein right to the area. These cells are readily sourced through biopsies, so the collection of them is quite straightforward. And it's the autologous approach, we believe, reduces problems with immune rejection concerns. And also, it's based on a proprietary platform of manufacturing we've developed over years and have been able to optimize. We are using a third-gen self-inactivating lentiviral vector. The reason for the selection of the vector, the gene construct is quite large, like 9 kb, so the packaging capacity was of consideration. Also, the integrative nature and the integration of dividing and non-dividing cells, we believe, can contribute to protein, longer in chronic disorders, longer protein expression, and we're interested in that as part of our programs. And of course, lentivirus has had a significant clinical experience, including in an approved CAR T therapy. So, we also are managing that as part of our process. And then together, we have these ex vivo cells that we, have release testing we can do for potency and look at transduction rates. And also, as I mentioned, administer intradermally. So this is the program, and we manufacture them in an autologous fashion. So we're collecting small 3 millimeter round biopsies from patients, and then we bring them to our GMP facility, which I will talk about on the next slide, in Exton, Pennsylvania, outside of Philadelphia. We expand these cells, and then we introduce them to the viral vector, as you can see. And then we bring them up into large populations in hundreds of millions of cells into billions of cells. And then these cells, after they're harvested, are cryopreserved. So once we create the initial bank of autologous cells for these patients, we purposely create a very large amount of them, because the idea would be we won't need to go back to the patient for additional tissue. We'll have this banked cell population for them to use over the course of either their lifetime or their treatment time frame. And once they order a cell treatment, it only takes a couple of weeks for us to prep and have them out to the site, which coincides with their scheduling of their visits, because in a lot of cases, these patients are highly debilitated, and it's not like an instant next day sort of thing. So we think we have a nice bank program for this. And we produce them in our GMP facility in Exton, Pennsylvania. And we've been very fortunate to have this asset for some time now. We have a 13,000 square foot manufacturing facility. which includes clean rooms and quality testing areas. We've had this since a program we had some years ago that was approved by the FDA. So this site has been FDA inspected multiple times and is now the manufacturing core for both of our FCX007 and 013 programs. So you can see a quick overhead map and a picture of our site there. We have four manufacturing spaces that we're sharing and doing multi-purpose use, so we're actually a multi-product facility under our INDs. We actually already have existing capacity to serve the U.S. market for RDEB. So what I'm trying to build here is the story that, as you've heard probably multiple times, that manufacturing is obviously a very core issue with scale-up of these products and eventual commercialization. So we felt that we had the asset, the investment into that early was very important. So, you know, that between that and our on-hand vector supply, we think we're in good shape through the clinic. And, you know, we leveraged a lot of our previous cell therapy knowledge putting all these programs in place early, including things like quality systems, logistical cold chain, you know, obviously a big challenge with cell therapy and gene therapy products, all of our tracking, traceability, and contamination and inventory control, you know, already in place from our previous experience. So we think we have a nice leg up on the CMC. So just to move into our lead program, again, in RDEB, you just heard a little bit about the disease, and I'll just touch on it for anybody new in the room. But again, you know, this is a very devastating, blistering disorder that affects primarily children, as you can imagine, because of the mortality rate. It is a monogenic disorder. The gene is called COL7A1. It encodes type 7 collagen, which in turn forms an anchoring fibril. And if you think of like Velcro, you have like sort of the hoops on top, and then you have sort of the lattice below. The hoops are like the anchoring fibril, and then when you stick them to the skin, they bond together, and then that keeps all of our healthy skin together. But in their case, they either have truncated or null hoops, and they don't form, and so therefore you have two layers of skin that are floating on top of one another. You get a blister, and then you break, and then you deal with chronic wounds. And as you can imagine, it's a terrible, painful disorder. High mortality rate, about 76%. In the, you know, by the time of the 30s, mostly squamous cell carcinoma, very aggressive in these patients, and sepsis. Currently, no approved therapies, mostly palliative home care, very expensive home care costs, not generally reimbursed to full, and we believe it's about 1,100 to 2,500 patients U.S. So we are in the middle of a phase 1/2 clinical trial program now, working with Stanford University. We also brought on Colorado Children's to work on this study with us. We're looking primarily at safety, but we're also looking at pharmacology endpoints, including immunofluorescence and IEM, looking at protein structures in the skin. I'll show some data that we recently posted, and also some wound healing endpoints. And the biggest challenge in these studies is that the wounds you select, you know, are they chronic, are they reoccurrent? So we put in this wound monitoring period up front for at least 4 months to monitor these wounds to make sure the healing isn't just natural body healing, it's really done by a chronic, and do we see activity of the product? So a lot of these were developed with our KOLs, really learning a lot about these wounds and the proper way to approach this. And I'll share a bit of data we reported at the International Investigative Dermatology meeting back in May. We, so far in phase 1 of adults, 4 adults, FDA asked us to start in adults and then petition them to treat children, which we've done and gotten cleared back earlier this year. 7 wounds, age range from 20 to 37, and wound sizes around 4 to 23 square centimeters. This was a single intradermal injection session. Again, we can do multiple injection sessions. We actually have done that in one of our patients to look at repeat dosing and the impacts on that, and we've seen favorable responses. And we did all these injections under conscious sedation or topical therapy. So, you know, not a surgical process, outpatient, but we do believe for pain management, there are options for these patients, and the conscious sedation actually was a popular choice and actually well tolerated. So we'll continue to use that process. And from a safety standpoint, you know, FCX007 well tolerated out to a year. We haven't seen any serious adverse events or product-related events. No replication competent lentivirus. RCL testing is very common when you're using these vectors, either in the product, the vector, or the patient's sera. So we do it all the way from manufacturing to the sera to make sure we don't see any issues. And then when you're doing protein replacement, autoantibody concerns could be a problem. So we have checked serotype of the autoantibody response to these and haven't seen any detectable amounts in the patients. You know, anytime you have a patient who has a null response to a protein, you know, there's always the concern of an autoimmune reaction, and we have not seen that so far in the trial. So a little bit on the pharmacology side, these are immunofluorescent that are stained with the anti-Col7 antibody in the middle. The top is laminin 332. It's its binding partner, so the top line shows you where in these 2 human patients you've had a basement membrane zone intact. And as you can see in the second line, these are 2 examples, 25 weeks, 52 weeks, and 4 weeks to 32 weeks of collagen 7 expression in vivo samples we collected from biopsies. And interestingly, it seems to be increasing in expression over time. So, we think we're seeing an interesting phenomenon of how the mechanism of Col7 formation works. And we also have seen that in our we also see that in immune electron microscopy. So in these cross sections, what you're seeing are the basement membrane zone here. This would be more like epithelial, and this is more the dermal layer. And the dark spots you're seeing are actually the NC2 portion of the protein. So what is that? And the collagen structure, as all collagens are, it's triple helix, and that is flanked by 2 non-collagenous regions. The reason why we're focused on NC2, it's the final manufactured portion of the protein when it's translated and transcribed. So in that case, we're seeing full-length collagen 7 production in these plumes here. And then, of course, we've also seen anchoring fibril structures in these patient subjects. So very encouraged by the pharmacology results we're receiving, and also thinking about clinical endpoints and efficacy that are important to these patients. We have looked at wound healing as one of our potential endpoints. And through a lot of the literature and work being done at Stanford University, there's been a lot of focus around healing these at least 15% to be clinically relevant for patients. And we looked at some of our healing rates at 4 weeks out to 52, and also looked at it at 75%. So as you can see, we've seen some very encouraging wound healing rates going out even, these are low ends here, so we're still tracking these, but even at our 4 and 12, compared to untreated match control. So the way we're controlling this trial is, not using inter-patient control, but intra-patient, similar size wounds. One is treated, one is untreated, and then we track them through all the endpoints and through all the follow-ups. So here you're seeing untreated wounds and what their wound healing rates look like, and as you can see, generally left open chronic wounds. So very encouraged by the wound healing portion here. And the idea is to continue these phase 1 patients all the way through their 52 time point. And then our second phase 2 is a 6-patient enrollment, including pediatrics. So now we're looking at patients 7 and above. And we've done a few things here. We've increased the dosing in our trial and actually added a second injection session based on some of the safety data. We want to see what repeat dosing will look like for these patients. And do we even see better expression, or do we see even more duration? We've already treated our first patient in June. And as of now, we have 5 out of the 6 enrolled. So 4 of which are peds. We've actually been very excited to see a lot of pediatric enrollment in our trial, a lot of interest from that front. And then we're looking to finish that one up actually in the short term. And we're looking to report out some more data on our clinical trial in the first quarter of 2019. Just recently, we were awarded a $1.4 million grant over 4 years. So it's a small grant, but from FDA's OOPD's Orphan Drug Division. So we were happy to have their support in our trial, and we'll continue to work with them as well as we go through the steps of the trial. So that's our company. Thank you very much for listening. And again, we have 2 programs in the clinic, one in phase 2, one just entering phase 1. Really want to emphasize our manufacturing capabilities, and that we'll continue to develop those as well, and then continue those for any future programs we're going to add on, and take advantage of our designations, as you've heard, I'm sure, plenty of talking with the FDA to keep the dialogue open on moving this as rapidly as we can. So thank you very much.